All right, this is the review for Chapter 21, Part C. I went over this slide with you earlier. Just want to make sure because we're going to focus on the CD4 cells and the Cd8 cells and the Class 1 MHC proteins and the Class 2 MHC proteins. So make sure you understand that a CD4 cell is activated by a Class 2 MHC protein on an antigen presenting cell. Dendritic cell is just a type of macrophage that's found in connective tissues. So, I mean, it's just like a white blood cell. It's a phagocyte, is what it is. It's a phagocyte that has engulfed a pathogen and is presenting just that portion called the antigen or the antigenic determinant that is going to be recognized by this CD4 T cell because the shape fits the receptor on the CD4 T cell. Now, once that activation happens, then the CD4 T cell can become a helper T cell, a regulatory T cell, a memory T cell. Okay? Now, if the T cell receptor, if it's a Cd8 T cell, it's only going to recognize Class 1 MHC proteins, and those will also be presented by an antigen presenting cell, but it's only going to recognize Class 1 MHC proteins. So we're going to need to talk about the difference between the two of them. But that Cd8 cell can differentiate and become a cytotoxic T cell, and it can also become a memory cell. Okay, Class 1 MHC proteins are displayed by all cells except red blood cells, and that is because Class 1 MHC proteins, remember MHC stands for Major Histocompatibility Complex. These are just proteins on the surface of cells that identify that cell. We have Class 1 MHC proteins that don't activate our immune system. They're called self-antigens. And then Class 2 MHC proteins are displayed by, all of these are cells that have engulfed something and are displaying its antigenic determinant on their surface. So Class 2 MHC proteins are displayed by only antigen presenting cells. Okay? So a Class 1 MHC protein is displayed by any cell that has a nucleus in it. Both types of Class 1 and Class 2 MHC proteins are actually made in the endoplasmic reticulum. So that's where they're actually produced. Okay, and the only thing here I just want to make sure you understand is that Class 1 MHC proteins activate Cd8 T cells. They can be self-antigens or they can be non-self. They can be foreign antigens. But they're Class 1 and they can be found in any cell that has a nucleus. Okay? So like, okay, so what's a cell that has a nucleus that can display a Class 1 MHC? Well, not a red blood cell, but a skin cell, a muscle cell, a bone cell. I mean, any cell in our body is going to have Class 1 MHC proteins on the surface of the cell because they're called self-antigens. But they're not going to activate Cd8 T cells because Cd8 T cells already know to leave self-antigens alone, except in the case of autoimmune disorders. Now, the Class 2 MHC proteins are CD4 T cells. Recognize those, okay? And those are only found on antigen-presenting cells, like they're either found on dendritic cells, macrophages, or B cells. This is a chart, and it shows you all the information I have just given you. Spend a little bit of time with the chart. These cells look like cuboidal epithelial cells to me. They're nucleated, so any type of cell that has a nucleus can display a class 1 MHC protein on its surface. Okay? And that includes an antigen-presenting cell. Antigen-presenting cells can also display class 1 MHC proteins. But if they're self-antigens, the Cd8 cell recognizes that and leaves them alone. It says it down here. If the cell is an APC, it says, I belong to self, but have captured a foreign invader. This is what it looks like. Kill any cell that displays it. If the cell is not an APC, I belong to self, but have been invaded or become cancerous. Kill me. So an antigen-presenting cell can display an MHC class 1, and an antigen-presenting cell an antigen-presenting cell can be, sorry, an antigen-presenting cell can display a self-antigen, but the self-antigens, it also displays foreign antigens, okay? And then any cell that's not an antigen-presenting cell can display MHC, class 1 MHC protein because it has been invaded or become cancerous. Okay, I'm sorry, I made that really complicated and I didn't mean to. Let's go back to what's really important. Class 1 MHC proteins can be displayed on any cell with a nucleus. So the only cells that can't display class 1 MHC proteins are red blood cells. Any other cell can. Class 2 MHC proteins are limited to antigen-presenting cells, specifically dendritic cells, which are phagocytic cells found in your connective tissue, macrophages, which are found everywhere in your body. They're just large white blood cells that engulf and destroy pathogens, and B cells, B lymphocytes. Okay, so let's see. Okay, I will go over this with you. Oh, sorry. Okay, so the way cellular immunity works, so you've got a dendritic cell, which is an antigen-presenting cell, and it's got a class 2 MHC protein on it. Because what happened was it engulfed a bacteria, and it took a piece of that bacterium. It took the class 2 MHC protein from the surface of the bacterium. It displays it. And then it also has a molecule on its surface called a co-stimulatory molecule. So this first phase is called antigen presentation. This dendritic cell is an APC. It's engulfed the bacterium and it's displayed or presented the MHC class 2, the class 2 MHC protein on its surface. Okay, that's step 1. Step 2 is double recognition. Double recognition ensures that it doesn't by accident recognize something that it shouldn't be killing. So it's like a double check on making sure that we need to kill this particular anything that contains this particular class 2 MHC protein on its surface. So the first part is that the CD4 T cell recognizes the antigen MHC complex. And the T cell receptor binds both to the antigen complex and that co-stimulatory molecule there. Okay, and then binds to the antigen MHC complex. Okay, sorry. And then the co-stimulatory molecules bind. So both of those things, the antigen and the co-stimulatory molecule, in other words, the class 2 MHC protein or antigen and the co-stimulatory molecule have to both bind to the T cell, the CD4 T cell, That's double recognition, which is just a way of ensuring that a mistake is not made. And then all of a sudden, the T cells, the CD4 T cells, start to proliferate and differentiate. They start to form memory CD4 T cells that are going to help if the infection comes back. And then they start to form helper T cells that are going to go on and help destroy this infection right now. Okay? I'm going to skip some of this. There is good information, but I don't have time to do all of it. Helper T cells can also help in humoral immunity. Here's what has happened. Before so this cell that we're looking at that's purple is a B cell, is an MHC protein, MHC2 protein of a B cell displaying that MHC2 protein. Class 2 protein, and this is a B cell, and this B cell has antibodies, produces antibodies, is capable of producing antibodies. Well, before it can release those antibodies, the helper T cell has to activate it. So the helper T cell has to recognize that class 2 MHC protein on that B cell. Okay? And once that happens, then the B cell is activated, it can start releasing antibodies, and we talked in the last review about how antibodies work. Now, Cd8 cells are activated in a similar manner. The helper T cell has to, in fact, let's get all of it. Okay, the helper T cell has to bind. Here, our APC is a dendritic cell, and it is displaying a class 2 MHC protein. And a class 1 MHC protein. So the class 1 MHC protein is binding to the Cd8 T cell, which is going to become cytotoxic T cells that are going to go and destroy other cells, other harmful cells. But then the helper T cell, before the CD8 protein the CD8 T cell can be activated, the helper T cell must recognize the class 2 MHC protein. So it says the helper T cell stimulates the dendritic cell to express the co-stimulatory molecules. And these molecules are called interleukin-2. The dendritic cell can now activate the CD8 T cell with the help of interleukin-2 secreted by the helper T cell. So until that helper T cell recognizes the class 2 MHC protein on the antigen presenting cell and gives permission, basically, by secreting interleukin-2, the CD8 T cell cannot become activated. So this is a way of keeping your immune system from going crazy and killing cells that shouldn't be. So what do cytotoxic T cells kill? They kill cells that are infected with virus. They kill cells that contain bacteria or parasites. They kill cancerous cells, and they kill foreign cells. In the case of if someone with a different blood type that doesn't, a blood type that isn't compatible is transfused into the patient, or they are given a transplant and they are having a transplant reaction. The way cytotoxic T cells kill is they release perforins and granzymes, and I just think about it as perforins are like bullets. I mean, they create pores, and then the granzymes enter and stimulate apoptosis in the cells they're trying to kill. So they just kill other cells. This is a good table that shows cells and molecules of the adaptive immune response, a very good table, table 21.8. And HIV destroys helper T cells. So basically you destroy helper T cells, then you shut down the immune system. And I'm running out of time, so good luck on your exam.